Okay, so back for chapter 8. Now, let me see if I can get this to actually talk to us this time. No, for some reason we can't get that to actually... Oh, here we go. That's better. Okay, so defending self. So last chapter we looked at the fact that we are surrounded by, covered in lots of pathogens that all just want to use us for food and shelter. It's okay for them, but what about us? And of course, we don't die. There's 7 billion humans on the planet, and we tend to get stronger and stronger as a species and are dying later and later. So clearly we have some system of cells, tissues and organs which deal with this problem of pathogens. And so this chapter is all about our immune system. What is it that allows us to not get sick all the time? Because let's be honest, we should be, with all the stuff that's around us. So, our immune system, I've just done it, haven't I? Our immune system protects us from all these pathogens. And the first line of defense is called non-specific. It's our skin. So I'm holding my hand, looking to myself. So our skin, let's see, where's the camera gone? There we go. Our skin actually creates a barrier to the outside world, to our external environment. So that means that we have epithelial layers, skin layers, around all of our exterior. So our hands, our feet, our arms, that's all what we know of as skin is part of this non-specific first level of defense. That's also true of the linings of our digestive system, the linings of our respiratory system, the linings of our urogenital system. Anywhere where there's an orifice that leads into the body cavity, there's a layer of epithelial tissue protecting us from pathogens. And so the first thing that a pathogen's got to do is get past that skin. And of course, I'm not talking too bad today. I had a working bee yesterday, but I've managed not to make too many holes. All the time we're doing things and creating holes in our immune system, which means, of course, that once we've passed that first line, we need something else. So, this first layer of defense is skin, and it was discussed with all those other orifices, leads to cavities, also have the skin layer on them. But also onto that skin we have secretions of chemicals. So things like lysozymes are found on the skin surface, and these are found in our sweat. And they are excreted onto the surface to try and help keep these pathogens under control. So essentially our first layer of defense is doing a pretty good job most of the time of stopping us getting ill. And you can see there's this nice, no, I've lost it, this nice cross section through the skin here. There's that epithelial dermis on top, and then there's a hole down the shaft there, of course, down the hair shaft, that sort of opens it up. Sweat pore offers an opportunity for getting inside as well, but these are pretty small. And then below here we've got plenty of fluid and blood bringing things into this area to stop further problems. So, what happens if you cross that first layer and get inside the body? And of course they do. We cut ourselves open. Eating food, you might burn your mouth. All sorts of ways of causing holes in that first layer of defense exist. And of course these things are looking for those opportunities. A bit like foxes trying to get through my predator-proof fence at school, looking for that weakness. So if they do, then they get met by a whole lot of chemicals and cells that are waiting for them. So you have cytokines. These are signaling molecules that indicate the presence of damage. So if there's a cut somewhere, then they trigger a whole lot of other cells that know that we've got a problem. We need to seal up the outer layer. Interferons, a type of cytokine. These are actually produced by virus-infected cells. So they tell the immune system that there's a virus in the body, and start flagging those cells that have been infected so they can come and be destroyed. There's a whole lot of complement proteins are known as. So there's an array of blood proteins which actually can kill cells that they recognise as not belonging, as non-self. So how do we know self from non-self? Well, think back to chapter 1 and we're talking about our wonderful phospholipid bilayer. And we have these lovely little phospholipids there making the double layer, protein channel for moving things through. There should be some cholesterol in there somewhere. And then there's this carbohydrate protein complex, often known as a multi-histocompatibility complex, a MHC, and some of them are about marking self. There's quite a few of them, and they have different roles, and we'll talk a bit more about that too. But one is just about saying, I belong. So all of my cells should have an MHC marker that says Gary. So my immune system can float around and check that all the cells say Gary. And if it finds a cell it doesn't, then the immune system becomes quite hostile and deals with that problem of a lack of self, and engulfs by phagocytosis and destroys these things. So that's all non-specific so far. They're not particularly smart. It's just stopping things getting in and destroying anything it doesn't recognize. And then we have our specific immunity. This is a part of the process which actually says, okay, I recognize this as an invading pathogen, and I've either seen it before or I've been introduced to it before, and I know what to do, and I can deal with it. So this we find only in the higher evolved animals. It requires quite a lot of specialized organs. So here there's thymus here, spleen, Peyer's patches down around the pancreas and small intestine, bit of parts of the large intestine, our bone marrow where we make blood. So quite a few parts of our system are set up to actually start recognizing non-self and knowing how to deal with it. We also have the lymphatic system, and so lots of fluid that is in our blood system leaves the blood and enters the interstitial fluid, that fluid surrounding all our cells. And white blood cells are very good at leaving the blood system and entering that interstitial fluid and searching for things that shouldn't be there. But of course they need to be regained somehow and put back into the circulation system. So as they move through interstitial fluid, they get taken up by the lymph system, and then they get brought back up through the lymph system to the heart. You see here that most of these come around the left subclavian vein and the right subclavian vein. They drain back into the heart at the thymus and back into the blood system, and out they go again. These are two indicators of disease. Sorry about that, we just had an invader, a helicopter overhead. Now I can't get the, there you go back. And so these become swollen and inflamed if we have lots of white blood cells moving through here, and our white blood cell count rises. When people talk about white blood cell counts, we should have a low white blood cell count. If we're healthy and our immune system is dealing with things nicely and everything's ticking along well, then we don't have a lot of white blood cells. If you're infected, then your white blood cell count rises as we start producing more white blood cells to deal with it. So, here's a whole lot of cells involved in this incredibly complex system. And we get this inflammation response. So if you think about it, you cut the back of your hand, and that whole area becomes warmer and swollen and brings fluid to it. So a lot of those cells are coming to seek non-self cells, so might be trying to get in to destroy them. And that inflammation is what allows all of that material to arrive there. So the localized blood vessels expand and allows more fluid to the area. And of course we bleed, we lose fluid through it. But that's deliberately trying to flush out anything we shouldn't have as well. So there's a huge range of cells here, and we'll talk about some of them in detail as we move through. But you don't have to memorize all of them. There's just so many cells in our immune response. It's a really incredible response. So we have killer cells. There's phagocytes. So they're white blood cells that specialize in engulfing and digesting things that are foreign, that lack self. They're constantly moving around your body. When they find something they know, and they're concerned about the amount of it, then the phagocytes are able to actually call up other cells and make copies of themselves really quite quickly. If we're very, very sick, we get very, very tired. And that's because most of our energy is diverted to making cells that deal with the invader. We also have things called natural killer cells, and they can actually detect our own cells that have been infected or diseased by viruses and bacteria, and actually go and destroy those to stop them spreading the disease. In fact, we saw that, we watched that on that lovely Woodleigh.TV animation of apoptosis that's extrinsic to the cell. So, when our external barrier is breached, then inflammation occurs. We get increased blood flow to the damaged area, increased permeability of blood vessels, and increased movement of white blood cells to the damaged area. To prevent blood loss, small arteries constrict. Blood platelets become quite sticky and fragile, and that's caused by some of these blood proteins we talked about before, and the blood starts to coagulate. And then, of course, new tissue will grow over the wound in time. So we get that scab forming, and in time it closes over. There's also fever, localized fever at the site, and this causes the pathogen growth to slow, and hopefully some of that heat might, in fact, denature the proteins within the invader. So the inflammatory response is really quite important. Although those of us like myself who suffer from hay fever know how horrible that is. We also have an adaptive immunity. So Macfarlane Burnet won his Nobel Prize. He was director of the WEHI for a long time, working out how our body can make so many of these antibodies. So we're carrying plenty of antibodies in our system, and we can actually clone and make new varieties quite quickly because they have very few genes involved in this area, and they can mutate quickly to make new copies of themselves and change their structure. There's a lovely little Woodleigh.TV video about Macfarlane Burnet's work too, so we'll have to watch that. So antigens are essentially self-markers. So the self-markers on invading material is seen as non-foreign. And so an antigen is an antibody generator. We will produce an antibody to match the antigen. So antibodies are molecules produced by lymphocytes, such as our T and B cells, which we haven't talked about yet. And they can actually bind with a specific antigen on the pathogen, blocking its attachment site so it can't attack our cells, and flagging that it needs to be engulfed. And a phagocyte will come around and clean up the whole mess. So T-cells, they're produced in our thymus gland. Well, actually, they're produced in our bone marrow. They are matured in our thymus gland, hence the T for thymus. And they have antibody-like molecules on the surface, and these will bind with antigens. There are helper T-cells, and they produce and release cytokines in response to an antigen's presence, and cytotoxic T-cells. And so when they're stimulated, they target the foreign and our own infected cells and cause them to lyse. They kill them, those we saw in that little animation the other week. B cells, these are formed in the bone marrow and in our spleen, and they're usually small, non-dividing inactive cells. They actually lose... I'm thinking of something else. When they meet specific antigens, they become very active and produce plasma cells and memory cells. And this is really important. This is the whole basis of immunity. Having had the disease, the plasma cell can create lots of antibodies to it, and the memory cell remembers. So second time round, you don't get it anywhere near as ill because your response is much, much quicker when you recognise these antigens. So there's Frank Macfarlane Burnet. He was the one who decided to develop this clonal selection theory and won himself the Nobel Prize. I should explain that before, so I'll keep moving because I'm running out of time quite quickly. And it's actually his diagram from his work of how he believes clonal selection theory to work. Plants, of course, lack immune systems, they lack most body systems, but they do need to protect themselves against disease. So, of course, they've got cell walls around every cell, and they've got to be breached first to get inside the cell. But they also produce lots of secondary metabolites. They produce chemicals that are made from their own metabolic waste, and these kill invaders. And, of course, we use these now as the source of most of our antibiotics and drug treatments and our pain relief. So they're also quite capable of storing really quite high levels of toxins in their cells, and often they'll actually lose a whole branch that's full of toxic material. Let's dump the branch to protect themselves. I'll just squeeze that in less than 15 minutes. I'll be back soon with Chapter 9.